Oh, good morning and welcome to today's seminar, exactly 11 Eastern time here. It's a pleasure to have Amanda Hulse Kemp today with us. Amanda is a computational biologist that works for the USDA and is based here at NC State at our university. Amanda graduated from Texas A&M in 2015, and she and her lab have been using genomics and bioinformatics to support plant breeders with different tools, particularly high quality reference genomes. And she's going to speak to us about how her work is supporting building the genomic selection capacity for public cotton breeding in our region. So Amanda, it's a pleasure to have you with us. I'm going to stop the share here. I ask people to shout out where they're joining from. And thank you so much for joining us, Amanda.
Hi, and thank you for having me. I'm happy to join today actually from a breeding station for citrus for USDA ARS and outside of Orlando today. So hopefully this goes well. So just to get started, what I would like to talk to you all today is kind of giving an intro to my lab, as well as the background in cotton and public cotton breeding programs, and then look at a couple of examples for interdisciplinary collaborations we have with cotton scientists within the lab. So we'll take a look at building genotyping platforms, working in phenomics, as well as taking a look at genotypic resources, where we can go from where we are now, and how we're thinking about getting there. So to start, to give you an introduction to my lab, we are a part of the USDA Agricultural Research Service, and the USDA ARS is really into finding solutions and improvements to problems that affect Americans every day from field to table. So you may not be familiar with USDA ARS, so I'd like to give everybody a background. We're really kind of built as similar to a university where we've got 15 national programs or departments, and there's about 690 research projects split into those different departments. We have about 2,000 scientists and actually 90 plus research locations throughout the US. So my unit is the Genomics and Bioinformatics Research Unit. We'll see that abbreviated as GBRU. We focus on data generation, data analysis, and data integration, and we're a part of the Southeast area. So you can see the Southeast area here on the right, and you'll see some important information along with these stars that I've indicated on the map. So my star is this purple location, and that's where we're located in Raleigh, North Carolina. So again, I'm Amanda Hulse Kemp. I'm a computational biologist in the Genomics and Bioinformatics Research Unit, and my background really focuses on building interdisciplinary collaborations, as well as working on polyploid genetics, genome assembly, and tool and development and integration of those tools into breeding programs. So my current lab is composed of 3 postdocs and 3 students, and I'd really like to highlight Grant Billings. He's a PhD student, and the focus in his project is working on cotton genomics. So he's contributed a lot to this work that we're going to see today. So the why in our lab is really looking towards building better products for consumers. So what we really try to do is we kind of follow this pipeline. We work to identify the problem, then build an interdisciplinary team in order to deliver products. So this would go something like, we need to develop a high throughput genotyping tool for cotton to allow predictive breeding. We would then build an interdisciplinary team composed of breeders, biologists, producers, as well as big data analysts. And our lab kind of fits in the middle of these groups. And by working together, we're able to deliver products like the cotton 63 SNP-K array that you can see on the right here. So that genotyping array is this little piece of technology, and we can use that for doing things like we recently published, looking at the outlook for implementation of genomics-based selection in our breeding programs. So let's think about where we are kind of with our cotton breeding programs. Not everyone here is experts in cotton, so just to give you a little background, cotton is the largest natural textile fiber crop. It's also an important producer of oilseed. In 2021, the US produced 17.6 million bales of cotton. A cotton bale is shown here on the left. This is a typical cotton bale, and I'd also like to point out this is Dr. Restrepo Montoya, which is a former postdoc in the lab, who's also done a lot of work in this direction. So cotton has a large economic impact, both in the US as well as worldwide. And it's hard to estimate the exact amount of economic impact, but it's been estimated somewhere around 500 billion worldwide. Importantly, about 95% of our world crop is this allotetraploid species, Gossypium hirsutum. Gossypium hirsutum is often called upland cotton. And if you know a little bit about cotton, you may have heard Pima cotton as well. So just to give you a background on what is the difference between those two, Gossypium hirsutum is shown on the left, Gossypium barbadense or Pima cotton is shown on the right. So clearly those are two separate species, and they have some different attributes that make them valuable. So the upland cotton has a very high yield. Pima cotton comparatively has a low yield, although they're vice versa in quality. So Pima cotton has quite a high quality and upland cotton is low. But when you get paid for yield, that means 95% of our production is the upland cotton type. And when you go and look at buying products made of cotton, if you just do a Google search and shopping for a cotton polo, on the left would be traditional upland type materials. And the average price that you can see here is about $6.79 per product. If you simply add Pima to that cotton search, it basically increases your price by 2.5 times. So that's kind of an example of the quality difference and how that plays into the products that we see as consumers on the shelf. So in the US, cotton production is primarily in what is called the US Cotton Belt. So our production in 2021 is highlighted here, and you can see that it's basically across the entire southern US. So this then implies that we now are going to have research locations kind of spread across those locations. What we're showing here is the location of public sector cotton breeders, as well as informaticians that work along the lines with those breeders. And the function of cotton public programs is kind of shown here. So you can see that we're really interested in integration of genetic diversity and integration, enhancing fiber quality, as well as seed quality for added value products, integration of disease resistance, performing basic biological research, and then in some cases also doing cultivar release in the non-transgenic or organic realms. A traditional cotton breeding program looks kind of like this, where we're going to make crosses and generate F1 individuals and then put them through a series of selection trials. This takes usually somewhere around 9 years, as you can see here, and it's just a crossing, testing, and selection, and kind of just repeating this process in order to get to our products that we are releasing from the public programs. So that really gets us to thinking about what the needs and the challenges are for our advanced programs. And what we're interested in is how can we use some of these advanced breeding technologies in our public cotton breeding programs. So everyone as breeders are probably familiar with the breeder's equation. And what we need to do is think about ways that we can adjust that equation to our benefit to increase gain. So we're interested in methods to reduce generation intervals, improving that genetic gain, using standardized genotyping to help us along that way, as well as advanced standardized phenotyping methods and data management systems to integrate multiple field trials. So these are things we really need to work on in our realm to get to advanced breeding technologies. And in the back of your mind, advanced breeding technologies in current days is really thinking about genomic selection. So when we talk about genomic selection, it's not as simple as just applying a technology. You really need a lot of foundational components in order to move into being able to use that technology. So these components We like to visualize them kind of as a pyramid. You can't really get to the top of the pyramid without having the bottom. So we're really resting on data management, genetic resources, integrated software, as well as phenomics and genotyping platforms to be able to use advanced technologies such as genomic selection. So thinking about our interdisciplinary collaborations and where they fit into the scheme of developing genomic selection, we'd first like to stop at looking at genotyping platforms. So our first case study is looking at the cotton SNP 63K array. In this interdisciplinary collaboration, we were able to develop an array for high throughput, high quality automated genotyping that now allows for genotyping from the plant to having genotypes at 38,000 single nucleotide polymorphisms within 3 days. So what that allowed us to do is start to look at characterizing our germplasm. So you can see here on the left a PCA of looking at different germplasm types. So our improved germplasm is in red. We have wild landrace material that's the same species is shown in green, and in yellow is other Gossypium species. So you can see that we are able to kind of very nicely characterize the diversity that we see. We see what we would expect to see, and that we see tighter clustering within our improved germplasm. And then as we get farther away from our improved germplasm, we're able to separate our individuals, and we see that increased diversity. And if we are to look at the structure of those materials, we really are able to see that we can very easily separate the improved materials from the wild materials just with using this technology. To date, there have been 172 studies that have been published utilizing the array. And what we were able to then do is look at the diversity between the materials that represent US lines versus materials that are coming from other countries. And as you can see, the US lines appear to basically cover the diversity that we see with the materials coming from outside of the US. For breeders that are working within the US and actually outside of the US, this is pretty valuable because our germplasm collection distributes a lot of material, and we can cover and gain access to most of the variation that does exist in our advanced lines. Next, we started to ask ourselves, if the US lines do contain that worldwide diversity, is there any clear pattern or separation between cotton individuals that may be potentially adapted to different regions? So there are 4 classified growing regions in the US shown here on the left. We kind of have an eastern region, a mid-south plains, and a western region, and those are colored on the right. And you can just plainly see that there doesn't appear to be any clustering happening with those individuals that have been characterized as coming from those regions. So there's a lot that you can take from this as a breeder. You can see that there could be the potential for a lot more specialization or adaptation of individuals to those growing regions, which would cause them to have clustering. And we do see that there is value, the breeders have found value by exchanging materials across those regions, which can lead to not having those very specialized clusters start to pop out, which we were able to evaluate with the array. A difficulty of the array thinking into the future is that it is an Infinium Illumina platform, which has actually started to shift their technology, causing the current array to be discontinued as of last month. So CottonGen, the cotton database, distributed a survey to assess the needs of replacing this technology. And it appeared that most individuals that are responding really wanted to keep the ability of using something like the cotton array. And the customers, I guess, had different thoughts on what we should do as far as making that available as far as price goes. But it was clear that we needed to develop a technology that will replace the array. So showing some results of the survey on the right, you can see most of the individuals that are replying are working in elite material, but there are people that are using the array for many different varying research situations. We also see in the bottom right that the large percentage of the people that responded are indeed using the array, as well as a number of other technologies. So what this has told us is we need to, as a group, develop another collaborative project where we develop a version 2 of the array. So we have been working on this, and it's likely to become available mid-2023. The good news for cotton researchers is this is likely to have decreased costs compared to the initial version, but it will still have the same high quality. So please look forward to that starting next year. The second collaboration that I'd like to highlight today is looking into the phenomics of fiber tip biology. And this is kind of a case study for an example of things that we are working with breeders to think about collecting data at a much higher data rate. So recent research had indicated that the presence of 2 morphologies exist in early fiber tip types. So you can see that depicted here in the figure where you can see pointed tip types, as well as some hemispherical tip types published by Graham and Hagler in 2021. So we have started working with the Hagler and the Lobaton group at NC State to come up with a way to develop methods to speed up the molecular work that the Hagler group was doing. So what we were able to do with this collaboration with engineers, molecular biologists, and data scientists is to develop a computer vision method for automatic classification and counting of those fiber tip types. So this will soon be coming out with Graham et al. In applications in plant science. Moving on to looking at genotypic resources, we're really interested in characterizing and evaluating the ability to use genomic selection in our individual breeding programs. So this is looking at population genetics in the South Carolina PD breeding program with Dr. Todd Campbell. Our project goals in this case, we're looking at identifying SNPs from the cotton 63K array, and then using GWAS to look for markers that were significant associated with fiber quality, both looking at HVI and aphis, as well as seed oil and protein content, yield and yield components in 14 different field trials. We then wanted to look at evaluating the ability to use genomic prediction to predict a line's performance using relationship between the lines, and how incorporating those GWAS results as fixed effects into the model would lead to a potential of better prediction values. So for this study, we worked with 80 different cotton lines that had been released from the South Carolina PD breeding program in Florence, South Carolina. We genotyped 1 plant from each line using the 63K array. Interestingly, we found that the heterozygosity had quite a large range in these materials. And we were able to extract 15,000 SNPs that were polymorphic within this data set and had a very high call rate. So the high quality data coming off of the 63K array was great for this study. You can see the data plotted on the right is all the raw data for looking at seed oil, seed protein percent, upper half mean length, and strength at the different locations that we evaluated across the different years. So the takeaways that we can see here is that HVI length, HVI strength, seed oil percentage are fairly consistent across the field trials, indicating that they should be more heritable. And the takeaway too is that the lint yield and the seed protein percent are not as consistent and therefore less heritable. Our first key finding with the GWAS hits is that they really seem to vary by field trial. So this figure here is showing how many GWAS SNPs were identified across the multiple field trials. So if a GWAS SNP was only found in 1 unique field trial, it will be shown on the left of each graph. And if something happened to be in more than 1 field trials, then we're plotting the number that it exists in. So what you can see here is that there are a few SNPs that were significant in more than 7 environments, and that a lot of the hits for yield trials specifically are kind of limited to the lower number of field trials where you're able to identify those. So this is something you need to be conscious of when you are testing your different field trials, that it may be beneficial to treat those separately in GWAS analysis, both separate and analyzed together, as some effects are likely environmentally specific and are masked when data is pooled. So you really need to be careful with the GWAS results for certain traits, as they will have minimal overlap between the trials. Additionally, we found that genomic selection can be used now for HVI length and HVI strength. So those fiber quality characteristics are very amenable to moving forward with genomic selection. So these plots are basically plotting the prediction ability across our different environments. And you can see that there's great prediction accuracy for length and strength shown here on the left. We have kind of a lower prediction accuracy for some of the others, but in general, we had the ability to do pretty high prediction accuracies. But overall, it really depends on what field trial or how much data you're putting into the estimate of your trait values. So we did see the overall, you can see is in the black box here in each of the plots on the left. And in general, if you look at the other environments versus the overall, if you're able to run this number of field trials, you always get the best prediction accuracy by using the combined mean. So this is important and giving us an indication that we really need to think about increasing our trial levels and getting to multiple environments in order to get an accurate prediction of what a phenotype is for each of these traits for our individuals that we're interested in. And extrapolating those GWAS hits to include them as fixed effects really does usually improve your prediction accuracy, indicating that the biology really does matter. This plot is showing the effect when including a single hit into the model. So taking all of the individual hits that we have, including them as a fixed effect, and then looking at what the change in the prediction ability is by including that SNP in the model. So you can see that there are some SNPs that have a negative effect on prediction accuracy, but overall, a lot of the SNPs actually greatly increase prediction accuracy. So you can see the tail of this plot. There are a number of SNPs that very greatly affect your ability on increasing the prediction accuracy. So takeaway one here is that always test your GWAS with field trials separately and pulled together. As we have indicated before, some effects are likely environmentally specific and are masked when that data is pooled. Be careful with your GWAS results. We really, again, did not find many hits with stable effects across locations. So this kind of brings us into thinking about what is the future of genomic selection. There have been a lot of studies that have looked at being able to use genomic selection. I guess a lot in this case would be 4, but multiple, we've now seen this happening in multiple publications where we do think that it's possible to initiate genomic selection in public breeding programs. So our paper is shown here. If you're interested in the results or the methods about the results that we just discussed, you can find those in the Outlook for Implementation of Genomics-Based Selection in Public Cotton Breeding Programs by Billing et al. And then we also have publications from CSIRO as well as USDA looking at the ability to use genomic selection. So for us as cotton researchers, we're really interested in where do we go from here and what can we do with interdisciplinary groups to move this ability forward to being able to use genomic selection. So we really need to think about having the ability to cost efficiently genotype thousands of individuals each year per program. So if you recall back to the number of programs that we have in the public sector, this is a large number of individuals across the US that we need to be able to genotype. And we do not have the current ability to be able to do that. We need to look at the assessment of haplotypes in each breeding program and making sure that those are covered. So if we're thinking about using low density genotyping, that we can accurately impute up to the materials that represent the breeding program. So if we're using that for genomic selection. We need to look at improving our inter-program collaboration and phenotyping because we have very valuable expertise in different areas associated with breeding and moving towards using integrated data management and informatics platforms. So as a group, we have formed a group that is working towards doing this and collaborating for developing genomic selection. What we are trying to do is shown here on the right and integrating our available QTL and haplotype information, using that to try to develop the next generation of breeding platform, which will allow us to get more information per data point at a very increased cost. So we can think about genotyping those large number of individuals that do exist in the public breeding programs. And moving that into an aggregated data management system where we can share across those breeding programs as well as to public and to publications. And then running some advanced large-scale genomic selection experiments in the field and testing those to make use of a practical application to make something similar to the cotton genomic selection guidebook. So we've been awarded a couple projects that we have just initiated in 2022 to work towards these goals. And in those projects, we're really going to think about these important considerations, which include field trials for experimental validation of genomic selection, increasing the size of those field trials and the number of entries to make sure that we can develop training populations to support movement into genomic selection, working towards rapid cycling or a breeding cycle of reduced time to say 4 years compared to the 9 that we had seen previously. But we also need to make sure that we guarantee a sufficient seed supply if we go through reduction of cycles to make sure that at the end we're able to get enough seed for doing what we need to do. And then enabling the connectivity between the data sets and data sharing and exchange. So in order to support this, we have been working on integrated platforms for breeders. I am the director of our Breeding Insight On-Ramp project, which is a USDA ARS initiative to jumpstart the process of integrating advanced breeding technologies into breeding programs. So what we try to do as a part of this project is really think about integrating omics-based tools as well as databases and phenotyping trials. So we have started working on developing databases built on the breed-based backbone, working with Breeding Insight to support our commodities that we have been collaborating with. So as I mentioned, I'm currently sitting outside Orlando, working with the Fort Pierce unit with USDA that's working on building citrus. So we have initiated a citrus base, and we have this for each of the commodities, including cotton, where we can keep our breeding program data and allow that to be exchanged in a straightforward format to work with our collaborators to move forward to having the ability to manage these larger scale field trials than we have been doing in the past. We really also want to move forward to being able to phenotype those larger trials. So we've been working on developing computer vision models for detecting fruit on the tree or bolls on the cotton plants, and then being able to integrate those with omics-based tools and developing those omics-based tools if we need to, such as the cotton array. So what I'd really like to point out is that these interdisciplinary projects are quite big initiations. So this is all of the people that had participated in the development of the initial cotton SNP 63K array. And you can see that that includes multiple public and private institutions, both in the US as well as internationally, and multiple different funding sources. So these are big efforts that lead to the ability and technology that can be used for moving forward for allowing things like genomic selection. And it's really essential for these collaborations in order to be able to move forward with these big ideas, such as moving towards genomic selection. So I know that we have a large number of comments that are coming in. So I would just like to say thank you to everyone today, for the organizers for inviting me, especially for USDA ARS for supporting me, as well as NC State and our many collaborators at the many institutions, as well as the funding agencies, which we wouldn't be able to do this research without, including Cotton Incorporated, USDA NIFA, and the National Science Foundation. If you wish to find us, you can find our website listed here. And with that, I think I would be ready to take some questions.
Thank you, Amanda. Yes, we have at least one question coming, but I wanted to point out that we have a pretty diverse audience today. Some people indicated that are joining not only from here, from places in the US like Nebraska or Clemson University, but also from Tunisia, Morocco, Kenya, Pakistan, Nigeria, Zimbabwe, Poland, Peru. So pretty diverse audience, and I like when this happens. Okay, thank you so much. We have a question in the chat from Don Jones saying that for seed oil prediction, would you advise a high number of environments or delete certain environments that mask results?
You know, some of these questions are a little bit difficult to answer with the current amount of data that we have. So the uniqueness of the data set that we were showing here is that we had a large number of year by environment trials. So we had a large number of data. I think that I'm not sure that you would want to mask results from certain environments. I think ideally what we would like to get to is a point where if an environment is not quite useful for our predictions, we would of course not want to run trials there. But it may be, as you indicated, that potentially an environment might be useful for something, say yield, but not necessarily informative with something like seed oil. So we may decide informatically to make some adjustments that would allow us to get the most bang for our buck. But I think that that's some research that we still need to do.
There's a question that to show again the articles about genomic selection or send by email. I think Yusuf, we're gonna post the recordings, and you can advance the recording to that page and get the references there, if that's okay. Another question coming from Sandeep: is it feasible to use genomic selection in line development?
So that's our real target here, is for actually applying genomic selection in the programs. So what we'd be interested in is trying to think about using genomic selection at early generation intervals, and then directly using that for what would get moved forward in the line development.
Mm-hmm. So a question from my side, Amanda, how much of the current cotton production is based on lines, or how much is hybrids? And also, how much of this work sort of spills into the private sector also?
So I guess there's a lot of components to your question, but in the development of lines, so the US and most of the other countries really are working towards inbred cultivar line release. Most of the hybrids, there are a hybrid market, and that's mostly done in the Indian region, as far as I'm aware. I think that they're really the only ones that are working on hybrids.
Okay.
And then what was the tail end of your question?
I saw at the end that you have Dow as a collaborator, but how much collaboration there is, and how much of the results are used by the seed industry?
So we work pretty closely with the industry to develop some of the genotyping platforms, and they actually contributed some of the markers that are included on the platforms. And I think that they are very interested in the results that we find associated with these things. So having a standardized marker set is really valuable to everybody in the community, both on the private and the public side, because we have a standard set of markers that can translate across the different lines. So I'm not sure how much they have used our results in their research, but I mean, they have let us know that they are paying attention. So if they find results or we find results, then they're going to pick that up and integrate that into their systems, which are proprietary, of course.
Yeah.
But they were interested enough that they continue to collaborate and be involved with the research that comes out of these standardized systems. So this is something that we are trying to work towards in other crop systems too, is really having these standardized resources that if you're trying to do things like meta-analysis, you can really leverage standardized information. So it's something to think about in your other systems. If you don't have standardization, to think about how you can increase that in your research.
Right, thank you. Candace is asking if you have tried to analyze characteristics of the environments that are not useful for prediction of seed oil. For example, average minimum maximum temperature during the production season.
So we haven't went into analyzing environments yet, but that's definitely the next line of research that we should look into. Historically, it's been a little bit difficult, I think, to pull that accurate information, but increasingly a lot of our field trial locations are installing weather systems in order to be able to track that, because historically you would have to go and grab information that may not necessarily be exactly pertinent to the field that you were trialing in. So there would be some error there. But I think as we move forward with the addition of different sensors and weather stations on research farms, that's definitely something we should move towards.
Question from Satish, how you deal with quality traits in genomic selection? I think you talk about length and strength, isn't it, of fiber?
So based on our results, the quality traits actually look highly amenable to genomic selection. So those will be our first push with actually trying to apply, is looking at the quality traits. So we're going to be planning on using those directly and using the prediction values for moving forward sets of individuals.
Okay. Fikret is asking, what is the major challenge to implement genomic selection in cotton breeding? Is it having a reliable and cost-efficient genotyping platform? That's his first question. There's another question, if you can answer that one.
So I think definitely you have to have a way to do cost-efficient genotyping to do genomic selection. Otherwise, you just can't do it. So that's the first challenge in our project, is identifying or generating a new method for doing cost-efficient genotyping. The current system that we have, the one when I'm talking about the past with the cotton SNP 63K, is actually looking at about $50 a sample, which when we're talking about genotyping in our programs, we're looking at needing to genotype about 3,000 to 5,000 individuals on a yearly basis. So that is clearly not fiscally feasible for doing at that level at that cost. So our challenge and what we've surveyed with the breeders is thinking about how do you get down to about $10 a sample. I think at $10, $15, that is feasible. And there are technologies now where we can do that. So the question is, what is the right technology for us? And that's the first thing that we're going to be answering, because once you have that, then you can do a lot of these bigger efforts.
A related question he asked, the second question, is if you think that changing the platform, genotyping platform for a breeding population will create problems.
I believe that the hypothesis is we think is potentially yes, especially if you're decreasing the amount of markers to get to something that is a cost-effective strategy. So you need to make sure that those markers capture enough diversity in your breeding programs. The advantage we have in cotton, as we saw in the PCA analysis, is that we don't have too much separation into unique groups of germplasm. So we should theoretically be able to kind of leverage that to be able to use a similar system across our different breeding programs. As a part of our upcoming collaboration, we're actually working with multiple programs, and that's one of the questions we want to answer is, are you able to use the same platform in different breeding programs? So we should have an answer to that soon.
Good. Yusuf is asking, could you mention the specific qualities in the Pima cotton that made it the product more expensive?
So the qualities that are really desirable in Pima are fiber length, as well as looking at the diameter. So Pima has a very fine diameter and a long fiber length, which makes it really nice for making textiles. So if you think about making yarn, you can fit more Pima fibers in a thread of yarn than you can with the upland cotton. So that makes it feel nicer if you touch it.
Is there a way that genomic selection could take into account non-additive genetic effects? That's a question from Wilfred.
I think yes, but you're going to have a more complicated model, which I'm not sure that we currently have the ability to kind of look into, but that's something we definitely should take a look at. We don't see a lot of, so we're not dealing with hybrids too, which simplifies our use in that case.
Right. Sandeep says, thank you for an excellent presentation. His question is, to what extent do the GWAS data and haplotype information help improve genomic predictions, considering we have consistent GWAS results across the environment?
So we've done some work now with imputing up with haplotype information, and it greatly increases your prediction accuracies. So I didn't show that here, but I believe it is in the publication if you wanted to go take a look there.
Okay. There's a question from Yusuf. Did the heritabilities have significant effects on the accuracy or validation of genomic selection of the traits shown in your presentation?
Yes, it's definitely easier to predict a more heritable trait. I mean, that's just an innate factor of the traits. So the things that are less heritable are going to have less prediction accuracy ability.
Right. Okay. Any other questions before we adjourn? Please post them in the chat. I'm gonna share my screen again. Oh, thank you. A lot of message. Thank you for your presentation, Amanda, and thanks so much, everybody, for your participation today. It's been a great seminar, Amanda. Thank you for accepting the invitation. Next week, we're going to have Dr. Mariano Alvarez talking about interpretable machine learning to improve performance in association, discovery, and prediction. Mariano works at Avalo AI here in the Triangle, and will be again at 11 a.m. Next Thursday. Thanks, Brandon and Tiago, for your help. And the recordings will be, when you receive the announcement for next week's seminar, you scroll down and you will have the recordings down there, okay? A lot of people have written to me asking when are the recordings are going to be posted. Tomorrow, you're going to receive the announcement for next Thursday's seminar. If you scroll down, the recordings for the previous 3, this one and the previous 2 will be there, okay? So thank you so much. You guys have a nice rest of the week, a nice weekend, and we'll see you next Thursday. Thanks, Amanda.